Imagine, for just a second, that you are holding an architectural blueprint.
Right.
But this isn't the plan for a skyscraper, and it isn't drawn on paper. It's actually a biological schematic that is, well, it's 450 million years old.
Which is just a massive scale of time to even wrap your head around.
It really is.
Mm.
And what you're holding is the foundational plan for the very thing you are using to process this audio right now, the vertebrate brain.
It's basically the ultimate origin story because, the brain doesn't fossilize well, right?
Mostly just soft tissue.
Exactly. We can't just dig up a Devonian-era skull and look at the tissue inside to see how human intelligence began. It's long gone. Instead, we have to look at living fossils.
So organisms that have barely changed over hundreds of millions of years?
Right. We use them to figure out how our own, highly specialized neural circuits actually came to be.
Okay, let's unpack this. Our mission today is to explore a groundbreaking 2026 research article from the journal Science.
A really massive study.
Oh, totally. The study is titled Lamprey 3D Single-Cell Transcriptomics Reveals Ancestral and Specialized Features of the Vertebrate Brain.
It's a mouthful, but the implications are wild.
So essentially, a team of researchers has taken this incredibly vast stack of data. We're talking single-cell sequencing, fully three-dimensional molecular atlases, cross-species comparisons, and they've used all of it to decode the brain of a lamprey.
And for those who aren't familiar, a lamprey is it's a jawless eel-like fish.
Right. But what we really want to understand today is why analyzing this specific ancient creature is the key to understanding our own cognition.
Well, to get to the why, we have to establish what exactly diverged 450 million years ago.
Okay, set the scene for us.
So that era marks the evolutionary split between jawless vertebrates, which the lamprey represents, and the jawed vertebrates.
And the jawed lineage is the one that eventually led to reptiles, birds, and, well, mammals like us.
Precisely. The lamprey is one of the most basal living vertebrates we have available to study. Its anatomy has remained remarkably conservative.
It doesn't change much at all.
No, not really. So by mapping the lamprey's brain today, we get a highly accurate baseline. It allows us to mathematically disentangle the ancient ancestral traits that all vertebrates share from the later evolutionary upgrades.
Like the heavily layered cortex that you and I possess.
Exactly. But before we can track those evolutionary upgrades, we need to understand the baseline itself.
Right. We need to know what the original, city grid of the brain looked like.
That's a great way to put it.
And the way these researchers map this biological city is fascinating because they didn't just slice up a brain and stick it under a standard microscope.
No, that wouldn't give them nearly enough detail.
Right. They use a combination of single-nucleus RNA sequencing and really high-resolution spatial transcriptomics technology called stereosec.
And that combination is really what makes this paper a landmark achievement.
Break that down for us a bit. What does that technology actually do?
Sure. So single-nucleus RNA sequencing allows you to look at an individual cell and identify exactly which genes are currently turned on or off.
Okay.
But traditionally, when you do that, you have to break down the tissue, which means you lose the physical location of those cells. You know what's there, but not where it came from.
Like having a list of residents, but no addresses.
Exactly. Stereosec solves that. It allows researchers to capture that highly detailed genetic data while preserving the exact three-dimensional spatial coordinates of every single cell.
So to put that in perspective for you listening, it's kind of like taking a highly advanced MRI of an entire city.
Right.
But instead of just mapping the structural outline of the buildings and the streets, which would be the brain regions, you're mapping the exact profession, the current activity, and the identity of every single person living in that city.
All without removing them from their homes.
Which is just mind-blowing. And the physical scale of this undertaking really cannot be overstated. The researchers took 40 consecutive slices of the lamprey brain.
And each one of those slices was just 10 micrometers thick.
So thin you can barely even comprehend it. And from those slices, they built a continuous, fully three-dimensional map.
Yes. In total, they mapped the spatial and genetic identities of precisely 465,778 individual cells.
Which is nearly a quarter of the estimated 2 million total cells in the entire lamprey brain.
It's a staggering data set.
Nearly 500,000 cells individually profiled. And from that data, they identified 14 major brain regions and 209 distinct cell types.
Right. And going into this, you might assume that a jawless fish from 500 million years ago would just have this primitive, unrecognizable mass of neurons.
Yeah, like a totally alien biology.
Exactly. But when they cross-referenced this ancient blueprint with modern mice, the foundational layout was actually surprisingly intact.
It shows incredibly deep evolutionary conservation, doesn't it?
It really does. The overarching topography is undeniably similar. When you look at structures like the thalamus, the hindbrain, and particularly the olfactory bulb.
The region responsible for processing smells, right?
Right, the olfactory bulb. They share remarkably similar spatial layouts and cellular makeups in both the ancient lamprey and the modern mouse.
That olfactory bulb finding is particularly striking to me. You are looking at a jawless aquatic animal, and it possesses the exact same stratified, layered architecture for processing chemical odors that a terrestrial mammal does.
It's wild, and the conservation goes all the way down to the molecular level.
Oh, wow, like specific genes.
Yes. The researchers tracked a specific gene called MIS2. Now, in mammals, this gene is absolutely essential for determining the fate of certain inhibitory neurons within the olfactory system.
So it basically tells those new cells what they're supposed to become.
Exactly. It's a regulatory gene. And in the lamprey, MIS2 exhibits the exact same conserved expression patterns.
So the molecular machinery that your brain uses to process the smell of morning coffee was essentially engineered, tested, and locked into the vertebrate genome 500 million years ago.
Yep. It's been running the same code ever since.
That makes a lot of evolutionary sense, though, considering that chemical detection smell is arguably the oldest and most fundamental way an organism interacts with its environment.
It's essential for survival across the board.
But this structural similarity only holds true for the basement of the building, so to speak, right?
Yeah, that's where things start to diverge.
Because as the researchers moved out of those foundational sensory and motor areas and mapped the forebrain and midbrain, that recognizable mammalian architecture just vanishes entirely.
We see a massive structural divergence when we look at those higher-order processing centers. Take the pallium, for example.
Okay.
In mammals, the pallium corresponds to the neocortex. It is basically the seat of advanced cognition.
And in our brains, the neocortex is famous for its strict lamination. It's organized into very distinct horizontal layers.
Like a highly organized filing cabinet.
Right. Or almost like an assembly line, where sensory data gets passed sequentially from one layer of neurons to the next. You have specific marker genes, like KCNIP2, that identify exact layers in a mouse brain.
Yes, very orderly.
But I imagine that assembly line doesn't exist in the lamprey.
It doesn't exist at all.
Yeah.
The orthologs, which are the evolutionary equivalents of those layer-specific mammalian genes, are actually present in the lamprey.
Wait, really? The genes are there.
The genes are there, but they don't show any discernible layer-restricted expression. They aren't organized into an assembly line. Instead, the lamprey's pallium features what the researchers describe as an inside-out arrangement.
Inside out.
Yeah. The inhibitory neurons are clustered heavily on the inside, and the excitatory neurons are on the outside.
Wait, if the pallium lacks those sequential layers, how does it actually compute complex information?
That's the big question.
Because an unlayered cortex seems like it would just be a chaotic jumble of signals.
And that is the core difference in how the ancestral brain processed data.
Mm-hmm.
Without the layered assembly line, processing is much more diffuse. It's less sequential.
Okay, so how does it work?
The neural networks in a lamprey operate on broader spatial gradients rather than the precise point-to-point cortical circuits we have. It is just a completely different topological strategy for interpreting the world.
And that alternative strategy becomes even more apparent when you look at the physical scale of the cells in the hindbrain.
Oh, the giant cells, yes.
Yeah, the spatial map revealed these massive structures, giant neurons and Miller cells that are over 4,000 square micrometers in area for a single cell.
They're absolute behemoths on a cellular level.
But from an energy perspective, building and maintaining a single cell that massive seems incredibly inefficient.
It is very expensive metabolically.
Right. So why expend the biological resources to build giant neurons?
It comes entirely down to the physics of locomotion in a watery environment.
Okay, explain that.
So action potentials, which are the electrical impulses that neurons use to communicate, they actually travel faster down a wider axon.
Oh, I see. So it's similar to how water flows with less resistance through a wider pipe?
Exactly. A wider diameter reduces the internal electrical resistance. The lamprey needs to execute incredibly rapid, undulating swimming movements to either evade predators or catch prey.
And those giant cells are powering that movement.
Right. These giant cells in the reticular formation express high levels of a gene called NEFM.
Which does what?
It codes for structural neurofilaments that literally physically support that massive cellular size.
It's the physical scaffolding for the giant cell.
Yes. And simultaneously, they highly express GUK1, which is a gene responsible for intense energy metabolism.
Because they need a ton of fuel.
Exactly. The organism is willing to pay the massive metabolic cost for these giant cells because that rapid electrical conduction speed is the difference between life and death in a predatory environment.
That physical adaptation makes a lot of sense. But if we move to the very top of the diencephalon, we find an adaptation that seems almost science fiction.
You're talking about the epithalamic complex.
Yes, which contains the habenula and the pineal gland. First off, the spatial data showed that the habenula is highly asymmetrical.
The asymmetry is profound. The left and right sides are executing completely different biochemical strategies.
Really? How so?
Well, the left side heavily expresses genes responsible for G-protein-coupled receptor signaling, while the right side is dominated by genes that regulate inhibitory neurotransmission.
So evolution just built two completely different processing centers sitting right next to each other.
Pretty much.
But here's where it gets really interesting. The pineal gland is what truly caught my attention.
It's a fascinating structure.
Because in human anatomy, the pineal gland sits deep inside the center of the brain. It acts as an endocrine gland, right? It pumps out melatonin to regulate our circadian rhythms and help us sleep.
Right. It responds to the light-dark cycle, but indirectly.
Exactly. But the spatial transcriptomics of the lamprey showed that its pineal gland is positioned at the top of the head, and it functions as a literal third eye.
And that is not a metaphor. It is an active light-sensing organ.
A literal eye.
Yes. The spatial mapping revealed a distinct laminar organization with a medial photoreceptor region. The cells in this region highly express the RBP3 gene.
RBP3, okay. What does that do?
In biology, RBP3 codes for a protein that binds to retinoids, which is a foundational mechanical step in translating physical photons of light into chemical signals.
But how does light even reach the brain through the animal's head?
So during its larval stage, the lamprey spends years burrowed in riverbeds.
Right, mostly in the dark.
And its primary lateral eyes are very rudimentary and often covered by relatively translucent tissue. So this pineal eye at the top of the head detects ambient light from above.
Oh, I see.
It signals to the animal whether it is safely buried in the dark riverbed or if it's exposed in the water column.
Wow. So it's a literal skylight for the brain to monitor the environment.
Biological skylight, yeah.
And what makes this so compelling is that researchers note our modern mammalian pineal gland actually retains a genetic echo of this ancient structure.
It's incredible.
Even though our pineal gland is buried deep under layers of neocortex and it hasn't directly seen a single photon of light in millions of years, the molecular footprint of that ancient photoreceptor remains.
It just repurposed it.
Exactly. It evolves from sensing light directly to receiving indirect light signals from the optic nerve to regulate our sleep.
It perfectly illustrates the broader biological rule. Evolution rarely invents entirely new structures from thin air.
It just relentlessly repurposes and modifies the existing architectural blueprint.
Exactly.
Which brings us to what I think is the most profound discovery in the entire paper.
The AENs.
Yes. Everything we've discussed so far is about how specific brain regions shifted. But the spatial transcriptomics uncovered a fundamental shift in how the individual neurons themselves operate.
Yes. And to contextualize this, the researchers didn't just look at the lamprey and the mouse. They performed massive cross-species integrations.
Right. They looked at a bunch of different animals.
They brought in newly generated spatial transcriptomic data from the zebrafish, which is a jawed but still ancient lineage, alongside existing data from macaques, birds, and reptiles.
A massive comparative analysis.
Huge. And when they analyzed the cellular identities across all these species, they isolated a massive population of cells in the jawless fish that they designated as AENs.
Which stands for enamioid-enriched neurons. Now, to grasp the significance of an AEN, we have to look at the strict rules governing modern mammalian circuits. In our brains, neurons are highly segregated by function.
They operate on a strict binary.
Right. You are generally either an excitatory neuron, meaning you release neurotransmitters like glutamate that stimulate downstream cells, or you are an inhibitory neuron, releasing GABA to dampen downstream activity.
You hit the gas or you hit the brakes.
Exactly. They are mutually exclusive identities in the mammalian cortex.
But AENs shatter that binary entirely.
They are functional hybrids.
Yes. The genetic profiling shows that they simultaneously co-express both excitatory and inhibitory markers.
I have to challenge this, though, because mechanically, how is that even possible?
It seems contradictory, right?
Yeah. You wire a circuit to send an on and an off signal at the exact same time. The circuit should just cancel itself out. How does a brain function if its neurons are hitting the gas and the brake simultaneously?
It's a fantastic question. Mechanistically, co-expressing both markers doesn't necessarily mean the cell dumps both neurotransmitters into the synapse in equal amounts at the exact same millisecond.
Okay, so what is it doing?
Instead, this hybrid nature gives the cell incredible local flexibility. It allows a single neuron to dynamically tune the network.
Like fine-tuning a dial.
Kind of. Perhaps releasing an excitatory spike followed immediately by a rapid inhibitory release to create a highly precise, tightly restricted window of activation. It provides a generalized flexibility to the neural network.
Okay, so the ancient brain was essentially populated by generalists.
That's a good way to look at it.
If we think of it like a business, the Lamprey brain is like an early-stage startup company. You don't have a dedicated IT department or a separate marketing team yet.
Everyone does a little bit of everything.
Right. Every employee wears multiple hats and does a little bit of coding, a little bit of sales, just to keep the company afloat. But as we evolved, the mammalian brain transitioned into a massive multinational corporation.
Where every single neuron has one highly specific, rigidly defined job description.
Exactly.
What's fascinating here is that that analogy holds up beautifully when you look at the evolutionary trajectory the data revealed.
Let's hear the numbers.
In the lamprey, these multitasking hybrid AENs are the dominant cell type across the entire brain.
Okay.
When you look at the zebrafish, they still make up an astonishing 94.48% of all neurons.
Wait, over 94%?
Yes. The vast majority of that animal's brain runs on these flexible, generalist startup cells.
That's almost the entire brain.
But as you trace the evolutionary tree toward more complex cognition, the architecture radically changes. In amniotes, the lineage containing reptiles and birds, the proportion of AENs drops dramatically to between 20 and 30%.
That's a steep drop.
And by the time you reach mammals like mice or macaques, they are almost entirely eradicated. In the mouse brain, AENs account for a mere 5.5% of neurons.
Wow. This implies that the core mechanism driving the immense processing power of the modern vertebrate brain wasn't simply adding more brain mass.
No, it wasn't just about getting bigger.
The mechanism was the systematic eradication of generalists. Evolution swapped out these flexible, multitasking hybrid cells in favor of highly segregated, distinct excitatory and inhibitory circuits.
Exactly. This shift was likely driven by millions of years of genetic duplications and mutations, which allowed for physical reorganization.
And once you separate the gas from the brakes into entirely different populations of cells, you can wire them into incredibly intricate, highly specialized networks.
That segregation is the prerequisite for the advanced, high-speed computation required for complex mammalian behaviors.
Which raises an interesting anatomical question. If mammals almost entirely abandoned these hybrid AENs, where are those remaining 5.5% hiding in a modern mouse brain?
They didn't just disappear evenly, that's for sure.
Right. The study points out that the remaining AENs are largely confined to one specific area, the cerebellum.
And that brings us to the final major mystery this paper tackled, because the evolutionary origins of the cerebellum have been heavily debated for a long time.
To understand why it's a mystery, we have to look at what the cerebellum actually does. In mammals, it is essentially a highly specialized, fiercely organized biological supercomputer.
Dedicated to precise motor control, balance, and learning complex physical movements.
Right. And it relies on a very specific architecture. A modern cerebellum has a complex laminated structure defined by the presence of Purkinje cells.
Those are massive, highly specialized neurons that process thousands of inputs simultaneously.
But historically, anatomists looking at the lamprey didn't see anything resembling that structure.
Right. Jawless fishes lack that laminated organization, and they don't possess mature Purkinje cells. So for a long time, the prevailing anatomical consensus leaned toward the idea that the lamprey simply lacked a true cerebellum entirely.
It seemed as though the cerebellum was a novel invention that only appeared after jaws evolved.
But anatomical structure can be deceiving.
Definitely. Because when the researchers applied their spatial transcriptomics to this area, when they bypassed the physical shape and looked purely at the genetic coordinates in 3D space, they discovered a cerebellum-like region hiding in plain sight.
They identified a specific cluster of cells that they designated as CB.1.
And what did the genes say about CV.1?
When they analyzed the transcriptomic profile of these cells, the data was undeniable. The genetic signature perfectly matched the profile of cerebellar interneurons found in the modern zebrafish.
Really?
Yes. Specifically, these cells strongly express SLC32A1, which is a classic marker for GABAergic inhibitory interneurons.
But the crucial difference is how they are physically arranged, right?
Right.
In a zebrafish or a human, these interneurons are meticulously organized into beautiful, discrete, functional layers. In the lamprey, these CV.1 cells are just diffusely scattered throughout the region.
Very disorganized by comparison.
So if they're scattered, how do they actually coordinate motor control?
They act as a diffuse processing filter. In a modern cerebellum, interneurons essentially function as the scaffolding. They inhibit and regulate the signals going to the star players. The Purkinje cells ensuring the motor output is perfectly calibrated.
Okay, that makes sense.
What this spatial data proves is that in the lamprey, this basic cellular scaffolding already existed and was functioning.
Even without the star players.
Exactly. The scattered cells still process sensory feedback and refine motor output. They just do it with far less mathematical precision than a layered network.
It fundamentally reshapes how we view biological development. Evolution built the supporting cast, the regulatory interneurons, millions of years before it introduced the star players.
It laid down the necessary molecular groundwork in the jawless fish, just waiting for later evolutionary pressures.
Like the need to navigate complex terrestrial environments.
Right, waiting for those pressures to organize those cells into tight layers and add the advanced Purkinje circuitry.
It perfectly encapsulates the overarching theme of the study, I think.
The building blocks for higher cognition were already present in our jawless common ancestor.
The incredible complexity of the human brain didn't arise from inventing entirely new brain regions out of thin air. It arose from the profound specialization, segregation, and spatial reorganization of very ancient cell types.
Just endless repurposing.
So, drawing all of this together, what does this all mean?
It's a lot to take in.
It really is. We began with the concept of a 450-million-year-old blueprint. And what this incredibly detailed 3D mapping of a jawless figure reveals is that the brain you are using right now, the neural circuits currently decoding my voice, is the result of an iterative, agonizingly slow upgrade process.
A really, really slow process.
Evolution retained the most effective critical systems, like the sophisticated molecular machinery dictating your sense of smell, or the light-sensing remnants of a third eye, while systematically dismantling the rest.
Over hundreds of millions of years, the vertebrate brain relentlessly swapped out those flexible, generalized startup neurons in favor of incredibly rigid, highly precise, segregated circuits.
We traded the innate flexibility of the ancestral hybrid cell for the sheer, raw processing power required to build civilizations.
Which, leaves us with a rather profound question regarding our future.
What's that?
Well, if the definitive theme of brain evolution over the last 450 million years has been this relentless march from flexible, generalized cells toward increasingly rigid, highly specialized circuitry, what happens next?
That is a challenging thought. Are we approaching a structural limit?
Exactly. As our environments change faster than ever before, driven by our own technological advancements, we have to wonder if our highly specialized, rigidly organized brains might eventually become too specialized to easily adapt to new paradigms. Have we as a species traded away too much of our ancient evolutionary flexibility in exchange for modern processing power?
It is definitely something to mull over the next time you find yourself struggling to adapt to a new piece of technology or trying to multitask at work. Just remember that 450 million years ago, every single neuron in your ancestor's head was a hybrid, built entirely for multitasking.